Hello everybody, welcome back. This is now third video of this first chunk of material. We're going to go a little bit out of order now with these learning objectives, just because it's a little bit, this is kind of where things kind of jump around a little bit. So we're going to start with this one down here. We're going to skip these two for right now, and we're going to kind of loop back and talk about those after. This should be a pretty short video. These two equations are pretty straightforward, and as well as this course doesn't require you to do any like big calculations. It's more kind of understanding the concept of these equations and what they're asking, or what they tell us. So we have two equations, the Nernst equation and the Goldman equation. Let me pull up the slides specifically for these. So, again, we're going to jump back into this a lot when we talk about kind of membrane potential as well. Like I said, we're going to kind of loop a little bit to get through everything. So, when we have a cell, there's lots of different positively and negatively charged stuff, both inside and outside of the cell. The big ones that we're going to talk about outside of the cell are sodium and chloride. Okay, sodium is positively charged, chloride is negative. There's way more sodium than there is chloride. Okay, so overall, the outside of the cell, let's write this down, outside of the cell is positive overall, okay? Because of how much potassium and stuff there is outside. Misspoke, sodium, how much sodium there is outside. So the outside of the cell overall is positive. The inside of the cell is very high concentration of potassium, and also it has a lot of our proteins. This A stands for amino acids. Remember with Biochem 1, we talked about how amino acids form together to make the polypeptide chain to then form proteins. So proteins overall are negative. So outside of the cell, overall positive. Inside of the cell, overall negative. Okay? So if you ask that exact question, reason why the inside of the cell is more negative is because of the high concentration of proteins within the cell. With this, we get something that's called an electrochemical gradient, right? Because all of these things have a charge, positive overall on the inside, negative overall on the inside. Positive on the outside, negative on the inside overall. And we know that positives and negatives attract each other, and then positives, negatives repel each other. So we look at this, so that's kind of the electrical component. So the outside stuff, a lot of this sodium here on the outside wants to come to the inside. Some of this negative stuff would like to more, the negative overall would want to go outside. That's kind of the electrical part of it. Main one we focus on is that the sodium. The sodium has an electrical force. Well, look at this. The sodium has an electrical force wanting to go inside of the cell. So that's the electro part. Let me write that down right here. We have an electrochemical gradient. The chemical portion is what we talked about with concentrations. Or before, when we saw that picture with diffusion, right? We looked at the different concentrations where they want to balance each other out. And this is dependent on each individual thing. So when we look at this, right, we have a whole bunch of potassium on the inside, not a lot of potassium on the outside. Even though we have other stuff on the outside, we're just going to look at potassium. So because there's less potassium outside than inside, chemically, potassium wants to go out of the cell. And then same thing for sodium. Because there's way more sodium outside of the cell instead of inside. Sodium wants to go in. So see how we have a chemical force of sodium wanting to come in, an electrical force of sodium wanting to come in, chemical force for potassium wanting to go out, and an electrical force for potassium wanting to stay in, because that inside is negative, and the positive and the negatives attract each other. So we get into something that's called the Nernst equation. The Nernst equation, this is looking at the membrane potential, or kind of the balancing of all of these forces, specifically for one single ion at a time. So if we wanted to look at potassium, for example, we look at the ECF versus the ICF. ECF is the extracellular fluid, so outside, and the ICF, which is the intracellular fluid. So we're looking at the outside, the fluid outside of the cell, comparing it to the fluid inside of the cell. We plug it all into this equation, and this is what pops out. We get a membrane potential for a single ion, and that is our membrane potential. Let me pull up real quick. A good definition for there's not a great definition for membrane potential. Oh, here's a decent one. So the membrane potential is the difference in electric potential between the interior and exterior of a biological cell. So we're looking at the electrical charge. Just like we saw here, we have that electrical force, those electrical forces, and the amount of charge inside and outside to give us a potential of each one. So that's the Nernst equation. It's for a single ion. That's the big thing we want to remember, is that it's a single ion. Now, well, we know we have tons of different ions, right? We have sodium, we have potassium, we have chloride, and there are a whole bunch of other ones mixed in there as well, like calcium, magnesium, iron, etc., etc., etc. So what we can do is we get this next equation, we take the Nernst equation and we can put kind of combine Nernst equations, and eventually spits us out this kind of equation. This is called the Goldman-Hodgkin-Cots equation. Biggest thing you want to remember for this one is that this is for multiple ions, right? So this single ion, this is the theoretical, and this is the real conditions in the real world. We have all of these different ions inside and outside of the cells, and this big old honking equation is what's going to give us the real membrane potential, the difference in charges comparing the inside to the outside of the cell. Okay, so this right here, these two equations, you don't need to really memorize the full equation. You're not going to be using the calculation. The kind of question that they're going to ask, they're going to focus on the single ion. Like the question would read, how do you determine the membrane potential for a single ion? And the answer would be you'd use the Nernst equation. They might write it out and give you a couple of options. So I would recognize the equation, the 61 over z times the log, and then also recognize roughly this equation as well, the 61 log, top versus the bottom, ECF, ECF, ICF, 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 ECF. Generally, don't forcibly memorize this entire thing and how to calculate all this stuff, but know what the equation generally talks about. Okay? That's going to be it for part 3. Just talking about those two equations and kind of what they or kind of what they refer to. Next video, we're going to talk about we're going to jump into the voltage gated, and we're going to look at the differences of the different at the gated channels. So we're going to look at the types of gated channels as well, and then the video after that will do the sodium and potassium pump.